Let's take a minute and talk about how a cell can move regions of DNA between the heterochromatin state and the euchromatin state. Now, in order to move a region of DNA toward euchromatin, there are two families of enzymes that are involved. The first family is referred to as SWI/SNF enzymes, SWI/SNF, SWI/SNF. These are a family of enzymes that are capable of rearranging the nucleosomes, essentially taking apart the carefully folded blocks of nucleosomes that are in heterochromatin and making them more euchromatin. The other family of enzymes are referred to as HAT enzymes, and the HAT stands for histone acetylation transferase enzymes. The name of this family of enzymes tells you what it's going to do. These are enzymes that add acetyl groups or acetyl groups to the histone proteins in the nucleosomes. Now, what happens when an acetyl group is added on to a histone is it removes some of the net positive charge that histone proteins have. Remember, DNA has a net negative charge. The net negative charge of DNA comes from the phosphate groups in the sugar phosphate backbone of DNA. So normally, the positive net charge on histone proteins and the negative net charge of DNA allows the DNA to be held very tightly when it's wrapped around the histones. By adding the acetyl group, the positive charge on the histone is removed, so the DNA just loosens up a little, it lets go a little. The combination of the SWI/SNF family and the HAT enzyme family is taking heterochromatin and turning it into euchromatin. Now, what if a cell needs the other thing to happen? What if a cell needs to take euchromatin and move it into heterochromatin? There's a different family of enzymes for that process, and that family are called methyl transferase enzymes. So these are enzymes that are going to add methyl groups to the DNA molecule, not to the histone proteins. These enzymes actually add methyl groups onto the DNA molecule, particularly in regions of the DNA where there's a lot of C's and G's. Remember, when we talked about the structure of DNA, we said that when base pairs hydrogen bond to each other, adenine and thymine can make 2 hydrogen bonds. Guanine and cytosine can make 3 hydrogen bonds. So guanine and cytosine are a little bit more tightly held together than adenine and thymine are in the double helix. By targeting the CG-rich regions of the DNA, these enzymes are targeting where DNA is a little tightly held together. These enzymes place the methyl groups onto the molecule, and the addition of the methyl groups makes it even more tightly held together as a molecule. Again, this is going to take chromatin from the euchromatin form and make it heterochromatin. Now, I mentioned at the beginning of the lecture that these global mechanisms largely focus on the area of transcription specifically. When we talk about gene expression, transcription and translation are involved, but most of the global regulatory mechanisms are around transcription. Once a region of DNA has been made into euchromatin, so it's looser and it's transcriptionally available, there are other ways that a cell can regulate whether or not transcription is going to happen. So just because DNA is in the euchromatin form does not necessarily mean that all of the genes in that region are going to get transcribed and translated. It just doesn't happen. There are other more fine levels of control. So imagine a region of DNA that is in the euchromatin form, so it's available for transcription, but the cell wants to control which genes get transcribed and which genes don't. There are a range of regulatory proteins that are going to do this work. These are proteins that are going to come in and bind to the DNA in the regulatory regions of the genes themselves. There are two big broad categories of these types of regulatory proteins that we need to be familiar with. The first kind are called repressor proteins, and the second kind are called activator proteins. I like the names of these proteins because they tell us what they do. Repressor proteins are going to be used by the cell to repress gene expression. Activator proteins are going to do the opposite. They're going to help upregulate gene expression. Now, both repressor proteins and activator proteins are going to bind to the regulatory region of genes, but they're going to bind to different places in the regulatory region. Activator proteins bind to what are called activator regions. That makes a lot of sense, right? But repressor proteins are going to bind to operator regions. That's unfortunate. Those two don't match, but repressor proteins bind to operator regions. Now, the promoter, remember, is part of the regulatory region of a gene. It turns out that operator regions are located downstream from the promoter. That's important because that's going to help explain how repressor proteins function. In comparison, activator regions in the regulatory region of a gene are located upstream from the promoter region. That too is going to be important when we talk about how these proteins work. Remember, both repressor proteins and activator proteins are literally interacting with the gene. So we consider their control as fine control. It turns out that by controlling whether or not transcription happens is something of a pile-on in the cell. First of all, the cell controls whether the chromatin is even available for transcription. Heterochromatin versus euchromatin. Of course, it has to be in the euchromatin form, even for transcription to be possible. But once it's in the euchromatin form, it doesn't mean that a gene is going to get transcribed and translated. It just doesn't. Because there are repressors binding to operators and activators binding to activator regions. There are also basal transcription factors involved in eukaryotic cells and sigma involved in prokaryotic cells, and whether or not those things can get access to the promoter. Remember, the initiation of transcription requires that sigma in prokaryotes binds to the promoter, that basal transcription factors in eukaryotes binds to the promoter. You can't even initiate transcription unless the promoter is accessible. I've written the word enhancer on this slide. Don't worry about that. I should have written activator. Listen, enhancer is a word that means the same as activator region. You'll see it in some source materials. But I'm going to refer to the region of the gene where activator proteins bind as the activator region. All of this means to us that there are several ways that cells have control on if a gene gets transcribed, when a gene gets transcribed, and how much a gene gets transcribed. There are lots of levels of control over that. That tells you something. That tells you that it is critically important that a cell have control over transcription. Proteins, it turns out, are very powerful things. And if a cell loses control over the production of protein, it can lead to the death of that cell. So cells invest a lot of time, a lot of resources, a lot of energy into regulating transcription. All genes are regulated just a little bit differently. Which control mechanisms are more active than others? It varies from cell to cell. It turns out that we know very little about how specific genes are regulated. Remember, we don't even know all of the genes that are in the human DNA molecule. We have an estimate. We estimate that there's somewhere around 30,000 genes, but we haven't found them all. And even among the genes that we found, we certainly don't know what protein they all code for. And even for the genes that we understand what protein is produced, we don't understand how all of them are regulated. There's still a lot of work to be done in this area. So let's talk a little bit more about this fine control of transcription of genes. Remember, a promoter is simply a region of DNA within the regulatory region of a gene that helps initiate transcription. It's the location where the RNA polymerase enzyme is going to bind so that it can begin transcribing the gene right at the first base of the protein coding region. Promoters are located upstream from the protein coding region of genes. Now, activators are also in the regulatory region of genes. An activator is simply a piece of DNA that can become bound to activator proteins in order to upregulate or activate transcription of that gene. As I said a moment ago, activator regions are located upstream from the promoter. Operators are different. Operators are pieces of DNA that can become bound with repressor proteins in order to inhibit or downregulate transcription. And operators are located upstream between the promoter and the protein coding region of the gene. So they are downstream from the promoter and upstream from the protein coding region of the gene. Now I have a term on this slide that says transcription factor. This word is kind of falling out of favor. This term is falling out of favor. We used to refer to all of the regulatory fine control proteins as transcription factors before we started to understand that some of them were activators and some of them were repressors. You will still see the term transcription factor in the literature. So understand that all a transcription factor is a protein that is capable of binding to DNA. That's all it is. It's a very generic word or term. When we use the term activator or the term repressor, we're being much more specific. Many activator and repressor proteins live in a cell in an inactive form. If you think about it, that makes sense. You don't want these activators and repressors to be active until you need them to be active. So cells keep them in an inactive form until they're needed. These are proteins, and one of the easiest ways to activate or inactivate a protein is to simply stick something onto it, even something as small as a phosphate group. Cells often add or remove phosphate groups from proteins to cause the protein to change shape and thus change function. So inactive activators and inactive repressors are present in the cell at all times, and by sticking a small molecule onto those proteins, like a phosphate group, cells can activate them and put them to work. Now, phosphate groups are only one of the things that you can stick to an activator or a repressor. There are other molecules that can activate these proteins as well. And they're often referred to as co-activators or co-repressors. They're simply molecules that can bind to the activator or bind to the repressor, cause it to change shape, and become active. Let's take a look at the diagram at the bottom of this slide. On the top, you can see an artist's depiction of a piece of DNA that represents a gene. So remember, a gene contains some code to build a protein, that's shown to us in brown, and some regulatory code, that's shown to us in blue. You can't see the promoter here in the regulatory region, but you can see an operator. Now, this particular gene can be turned down or downregulated or turned off, if you'd rather, by a repressor protein. But that repressor protein normally exists in the cell in an inactivated form. As long as it's inactive, the RNA polymerase enzyme can come in, it can bind to the promoter, which is up here somewhere, and it can start transcribing this gene. But when a cell wants to prevent transcription from happening, it can turn this repressor protein on. It does that by having this little small co-repressor bind to the repressor protein and cause it to change shape. Once it is changed in terms of its shape, it can bind to that operator region. Now remember, a moment ago I said that operators are located downstream from promoters and upstream from protein coding regions. So the promoter is up here somewhere. The operator is downstream from the promoter. The protein coding region is here, so the operator is upstream from the protein coding region. And this matters because look what happens when the activated repressor protein sits on the operator. It's going to bind to that operator, and it will physically prevent access to the promoter next door. When this RNA polymerase enzyme tries to come in and bind to the promoter, it can't. It can't do it because there's not enough room. Over in this side, of the diagram, you can see that in order for the RNA polymerase enzyme to bind to the promoter, it has to have room. It's going to overhang this operator region. If something is bound to the operator region, this RNA polymerase enzyme can't bind to the promoter. Transcription is being prevented, or it's at least being turned way down. There's another really good example of how repressors work that comes from bacteria. This repressor system is referred to as the tryptophan repressor. And again, it's a really nice example of how transcription is regulated. In the diagram on this slide, the double helix of DNA is represented by this red molecule. The repressor protein is represented by this green structure. And the co-repressor is represented by this little red molecule right here. Now, in this example, the co-repressor happens to be tryptophan. Tryptophan is just one of the amino acids. The gene to produce tryptophan is located in this area. So, if there's lots of tryptophan around in a bacterial cell, there is no need to transcribe and translate the tryptophan gene. We don't need any more tryptophan produced. We have plenty of it. So watch how the bacterial cell turns down or turns off this gene. When there's a lot of tryptophan around, that tryptophan is going to bind to the repressor protein and cause it to change shape. That will activate this protein. When it's activated, it will be able to bind to the operator region of the gene and turn that gene off. Now you might say to yourself, well, hold on, I thought genes were code for proteins. Tryptophan is an amino acid, it's not a protein. The gene here, or I should say the genes here, code for proteins. They code for the enzymes that are going to build the tryptophan. So when there's plenty of tryptophan around, the cell does not need to make the enzymes to build tryptophan. That would be a waste. That would be a waste of resources and a waste of energy. So at least temporarily, these genes need to be downregulated. And the best way to do that is with this repressor protein. When there isn't enough tryptophan around, that means there will be less binding of the tryptophan to this repressor. And if there's less binding going on, that repressor is inactive. It won't bind to the operator. The genes will be available and they'll be transcribed. It's important to know that the regulation of gene expression in living cells is very complex. We've so far only been talking about how transcription is controlled. But transcriptional control is only the beginning. Remember, transcription is the process that takes a DNA molecule and uses it to build RNA. But there are lots of other things that have to happen before you get an active protein. In a eukaryotic organism, the RNA has to be processed. It then has to be transported out of the nucleus so that it can be translated. And it's going to be translated only as long as it's not degraded. And the mRNA that gets translated and produces a protein is going to produce a protein that itself has to be activated or inactivated according to how well it's folded and so on. So each one of these steps in the production of protein gets regulated by cells. The control of transcription is critical, but it's only one spot in the pathway to producing protein. It's only one of the ways that cells regulate the expression of their genes.